Welcome to Science with Gurleen and Uyen. On this episode, we're going to be learning about protein synthesis.
But who cares?
Well, you should care because proteins determine the structure and function of cells, which ultimately determine you. Duh.
Okay, this is a chromosome. A chromosome is nothing fancy, just a bunch of DNA wound together. A gene is a section of DNA used to make many things: rRNA, mRNA, tRNA, but for now, we're just gonna focus on one of these, and that is the mRNA. Well, first, this section of DNA is made up of two helices, the 3' to 5' one and 5' to 3' one. The 3 to 5 strand is chosen as a template to make the messenger RNA in a process called transcription. What happens is a helicase enzyme unwinds the DNA. Then the RNA polymerase brings in nucleotides that contain bases complementary to the template strand. So adenine will be paired up with uracil because it's an RNA, and cytosine to guanine and vice versa. Every triplet of bases on this mRNA is called a codon. This mRNA then moves outside of the nucleus through the nuclear pores. This initiates the process of translation. Its first step is called initiation, a process in which the mRNA hooks up to the ribosome. The ribosome is made up of proteins and rRNA and is consisted of a large and small ribosomal subunits. Its job is to translate the codons on the mRNA to an amino acid. An initiator transfer RNA pairs with the mRNA start codon, which is AUG at the P site, and that anticodon is complementary to the codon on the mRNA. The ribosome moves and continues to read the codon series, and every time it reads the codon, a tRNA is called over. The tRNA carrying the first amino acid is already at the P site, and a new tRNA carrying the next amino acid in the chain is now arriving at the A site. The amino acid of the previous tRNA then moves on top of the new amino acid. This whole process of amino acid chain growing is called elongation. Elongation continues until a stop codon is reached. A polypeptide chain is released and the small and the large ribosomal subunits dissociate. The polypeptide chain from here goes to the Golgi body for further processing to eventually become a functioning protein. This process is known as termination.
So class, this was the process of protein synthesis. I hope y'all enjoyed the lesson. Please remember to like and subscribe this video. Peace.